don't want to sell it. It's been a case now.
Did you ever have that on the list? It was one of the and I didn't say.
Alright.
Class Trump was the best.
This is literally just sit there and
No, that's why I actually done the P50, the affinity one.
It is. It's on. We're live.
Stop.
No.
Stop. Stop. You're making me laugh.
You're. What's up?
Okay. So far, we have 2 people. Can you guys hear me all right?
It's good. You heard that?
Hey guys, can you hear me all right? Oh, perfect. Sorry about that, Leslie. I just saw that you responded. Okay. So I'm going to get started by asking if you guys have any questions. So the people who have specific questions could get started, and you guys could move on if you only have a small thing that you want to discuss. But if you have a more broader topic that you want me to review again, then I ask that we save that for a little bit later on in this live stream. Also for later, I shouldn't say as much, just because I did the review for that and we did it in class another time. So I feel like I'm not going to waste your time on that unless you really need to, and then we'll save it till the end for that. Do one of you guys mind going on the stream and just letting me know what questions are coming up, just so I have an idea?
All right. You doing that?
So that I don't have to check every 3 seconds.
Got you.
Yeah, I did. Oh, does someone ask that? Yeah, it's through email. Okay, so far, can you flip the screen?
Yeah, it is flipped.
All right. Just give me a sec. HDMI video mirror off. Now? No? Yes?
This Wi-Fi is not happening. Yeah, it's good now.
Okay. Alrighty, thank you guys. And it looks like the volume levels are pretty good. Let me know if I'm screaming or if I'm too low. So just let me know if you guys have any questions. So Brianna, Malia, if you have anything specifically, or Leslie, that you want me to answer, we'll get started with those. And then you guys get special treatment because you stayed here. So you tell me if you need anything specific.
No, I see it. Here.
What happened?
The Wi-Fi is so fast that it's not loading.
No way.
So, it's the same thing. Yo, he passed by like 5 minutes ago.
What happened?
And then the live. Showing like 10 minutes after.
Really?
Oh my
Oh,
God.
that's annoying.
Yeah, it's really
You just stood up there right now. Look, you
Oh,
saw
I just did that. Okay, that's not that bad. That's like 2 minutes. Okay, so people that are here, how can I help you? Or are you here for whatever happens?
Wait.
Okay. I'm gonna give them a chance to try and figure out what they want me to do.
No, can you just talk about the respiratory system?
Talk about the
Well,
rest.
I know it's a lot, but like
I would say blood.
the.
Especially if you only have a question you want me to answer.
Leslie says, can you put the O2 cascade graph that you ran in class?
Yeah,
Can I
let's.
tell you the questions that people ask?
Yeah, please.
Oh, you gonna say that?
I can say it.
Okay. So let me connect my laptop to here as well. And that's duplicating. Perfect. And now I'm gonna look up oxygen cascade. Okay, I'm pulling it up right now. I'm debating if I should give it to you. Oh, this is actually perfect. This is what we'll use. Okay. Okay, let me switch to it right now. And I just want to make sure that you'll be able to hear me when I switch. Okay, should be good. Can you guys hear me?
Yeah.
Okay. So, okay.
Oh, he put up the graph.
Okay, so let's trying to see if I can zoom. Oh, nice. Perfect. Okay. Alrighty. So I'm gonna use my mouse to do this, just so that you guys can see what's happening, and then I could switch back if you want. So first of all, when we're talking about the oxygen cascade, are you guys seeing what I'm talking about?
Yeah.
Okay, cool. So the whole point of the oxygen cascade is that air is going to keep moving from a place of high pressure to low pressure. So what you're looking at is basically a downward slope, or almost like a staircase down, that oxygen is going through this process as it enters your body. And the driving force for it entering your body is the fact that there is a gradient where it's going from higher oxygen levels to lower oxygen levels. Meaning the reason why you get diffusion across the respiratory membrane from the ambient air to the alveolar gas, and therefore alveolar blood soon after, is the fact that you have a decrease in the concentration of oxygen, or the partial pressure, because it's a gas and a mixture. And then you keep on going through that pattern as you go from the arterial blood, then to the systemic blood, and finally to the tissue that needs it. And the tissue is actually going to use it for aerobic respiration, that you're going from a place of highest down to the lowest. And that's why you have convection happening between ambient air, alveolar gas. That's why alveolar gas, arterial blood. Why convection from the arterial blood to the average systemic capillary blood, between that blood and the cells that oxygen. So another thing to note is the different units of the partial pressure that you're seeing. So remember how I said that you could say that pressure, which is the total pressure in a system, in the air that we're breathing, is going to be equal to 1 atm, or 760 millimeters of mercury. And from Dalton's rule, that you have when you have a mixture of gases in a total system, that if you add up their partial pressures, it's going to add up to the total pressure. And we know from Henry's law that in fact it's proportional, so that the more of the mixture that you are, the more parts of the mixture that you compose, the larger the partial pressure will be. So therefore, you're saying that concentration of a gas is going to be proportional to the partial pressure of the gas in a mixture. The reason why I'm bringing that up is that you could look at the left axis and you know that oxygen is 21% roughly of the air that you're breathing in. So when you look at the left side, you know that that's an ATM, because 21% of 1 ATM is going to be 0.2. On the complete right side, you know that that is in millimeters of mercury. Because if you take 0.21, so let's say roughly 1/5 of something, 20%, 1/5. So if you take 1/5 of 760, and we'll round that out to 750. So now divide 75 by 5, and you end up with about 150. So what that means is that your 0.21 ATM is actually your 160 millimeter mercury, roughly, of oxygen. And we got an O in the room. I'm very happy about that.
Can you repeat one more time?
Yeah, what's up?
You said something about the Henry's law. Like, can you repeat what you said about Henry's law about being proportional and
Right. So the concentration of a gas in a mixture of other gases is going to be proportional directly to the concentration of that gas. Meaning, more oxygen, higher pressure. Direct relationship. Meaning, as concentration increases, pressure also will increase. Which is why in the hemoglobin dissociation curve, that's why on the x-axis, when we're looking at PO2, we're saying that you would want a lower concentration getting the same binding ability. That would be cool for loading, you know, and that's why we're able to use that x-axis as not just a partial pressure, but also as a measure of your concentration, or an analog, a proxy. Okay? Any other questions right now? Okay, can we go over the lymphatic system, specifically the difference between adaptive and innate immunity, as well as what is opsonization? For the immune system, so really there's only very little that we covered. So we covered just about as much for the lymphatic system, and it's really about the function, which is to protect your body from pathogens for the immune system. And then for the lymph system, it's for both maintenance and surveillance, but also more importantly for the recycling of the volume of your circulatory system that is lost when it's crossing through the capillaries under high pressure, with very little circulatory system that is lost when it's crossing through the capillaries under high pressure, with very little vessels that are delicate So they're going to burst open sometimes and leak. So that 10% of it that leaks is then collected by your lymph system, and you have a lymph vessel that it basically has valves on it. And as soon as it fills, it's surrounded by a volume that creates enough pressure, you have a flow into that lymphatic vessel, and then that will then drain through your lymphatic system, through your thoracic duct, into your heart. Now, I'm saying more details now than what I said in class. And the whole idea right now is that you understand that this fluid that's being lost from your blood vessels is being picked up by your lymphatic vessels, and then it's going to drain back into the venous circulation and be part of your circulatory system again. So you're recycling the blood that has been lost through leaky capillaries. Again, that's going to be mainly the focus that I'm going for. The functions of it and the fact that we have these lymph nodules, which are small nodes, just like a venule is a small vein. And then we have immune function in the lymph nodes, which are acting like our TSA agents. And they're basically screening the lymph fluid that is going through the, or chyle, that's going through your lymph nodes. And it's saying, is there an antigen that's presenting that is not one of ours? So if it finds something that's a pathogen or something it does not recognize as self, it will basically have the B cells that are naive, that are in your nodes. Those will basically start to mount a response by opening up the door of your pathogen, meaning you have a binding of the antigen to the specific ligand that it needs from the B cell. And then that naive B cell will now become a memory B cell after that primary exposure by a specific door. And then also at the same time by creating things that will be able to attack your plasma cells. And they're gonna indirectly attack by allowing your T cells to then recognize things. So you have different types of adaptive responses. We don't have to know that for lab. So Brianna, did you need to know what opsonization is for lab or for lecture, or you're just curious? I'll wait for the answer. So in your book, if you want to flip over, I found this super cool diagram that kind of breaks down the difference between the adaptive and innate immune responses. And this is one of the simplest explanations I've seen. So let's see, where is it? Innate and adaptive work together. Okay. So Crash Course mentioned it, so I wanted to know if we needed it for lab. No, so you don't need it for lab. Opsonization, agglutination, complement system. All of these are different ways that you can have your adaptive responses, but it's not important for us. Opsonization is going to be a very specific way that you'll have an immune response. And basically what's happening is that you are marking a cell for destruction later on. So you have, so one second, let me pull up the page that it would be on. Where is it? Right. So whenever you have complement activation, part of it is that you'll have enhanced phagocytosis, meaning you're going to have cells the phagocytes, which are basically your big Pac-Man of your immune system, they eat things, they phagocytose them, which is literally they just swallow it. So those are going to get a message based on the opsonization to say, this is my target. So rather than waiting to see or looking around by patrolling for someone that's doing wrong, it's like someone called the tip hotline and said, this is the guy you're looking for. They gave a description to the police. So now the police aren't just looking around to see if anyone has a weapon. They're looking for a specific person that might have a weapon. So that's the process of optimization. But again, that's not going to be part of what I'm doing with you guys. Okay. For immune system. Okay, so back to what I was saying, that there was a page that I wanted you guys to look at. So if you go to where is it? I think I just passed it. So for the immune system in general, okay, I found the page, by the way. So on edition 3, it's on page 786, and it's entitled Innate Immunity and Adaptive Immunity Work Together to Defeat Pathogens. So I'll show you the diagram in case you're following along with a PDF instead. So this is what I'm talking about, this diagram over here. Okay, so what this is showing you is kind of the breakdown between the differences, different types of responses that you can have. And so if you're curious and you want to know for lecture, that's where you would be checking. Now, for me, what I want you to really understand is that your innate immune system is more broad and widely activated, and it's not going to be specific. So it's not going to be quite as effective. The cell types that make up your adaptive immune responses, because those, it's like getting a tailored suit. They're specifically made for that person. They're looking for someone, and this response, they know this is their weakness. This is who I'm looking for. This is how they look like based on their surface proteins. So they know what they're looking for, and they're adapted to have the most efficient response possible. But what that means is that they have to have time to train. So your adaptive immune system is something that develops with age, and they have to be, your naive cells have to be trained, and the way that they're trained is by primary exposure to something. So once you have your memory cells see an antigen that they don't recognize, they're gonna have first exposure to this, and this is gonna be slower. And in this time, they're building the specialist team that's going to be able to combat this very specific group of villains, right? So you have this enemy that has now come, and you want to make sure that the response is specific to them. So the way that you do this is by, like what I was saying again, you have a naive cell becoming a mature cell that has a specific key for a specific lock. But your innate system is going to be able to react to a lot more diverse things, and it's going to be more broad scale. Meaning, what's an innate immune response that we discussed in class? Something very, very broad that we're not targeting anything specifically. It's just let's see what happens. So the opposite of cold.
Happy. Not cold, hot.
Okay, so for those of you who didn't hear, she said the opposite of cold is happy. I'd agree.
First thing yell.
All right, so you have a fever, and a fever is going to heat up your body with the hopes that you're going to have an immune response that kills off the invading bacteria or virus, or causes through denaturing of its proteins. So proteins have a specific range in which they can operate and an optimal number that they want to be at. And if they're outside of their range that they can operate at, then something called denaturing can happen. And what that means is that it has a specific nature, right? Like when it's naturally looking like something, a protein has a specific shape. It has a structure. And that structure is going to become denatured. It's going to lose its natural appearance. It's going to look abnormal because the shape has the bonds that make up the shape that you're looking at are going to be destroyed by the energy that you put into the system. So you can use the energy of heat to break bonds that are stable. And remember that bonds, basically what happens in a bond is you have one high energy thing, another high energy thing, they come together and they both become more stable, which means they both lose energy. In order to break a stable relationship, it takes a lot of outside energy. The heat provides that energy to break down those bonds. And once you have the bonds broken down, now we are talking about the denaturing of the structure. So you end up with losing its conformation. The protein is going to lose its conformation. And since it loses its conformation, it loses its function. So fever is an example of one of those broad responses that your innate immune system has. And then you have a whole bunch of other ones. And if you're looking at this graph, you'll notice that you have your macrophages that are basically could attack, and those things are just kind of waiting for anything that they don't recognize to phagocytose it. But you also have, and then lastly, your adaptive immune system, like the name suggests, requires a more specific target, and first exposure will be slower while it's learning how to deal with this enemy. Second exposure, much quicker. This is why we give vaccines. So vaccines are a weakened form most of the time, or an inactivated form of the pathogen that you are trying to become vaccinated against. And the whole idea is that if you give it in this weak form, you will build an adaptive immune response so that once you get a second exposure, you get this quick, robust response, and it won't kill you, which most of these vaccines we give because they prevent vaccine preventable deaths. Your measles, mumps, rubella shot, and other ones. Could you please go over respiratory capacities, which include, okay. Did you guys have your questions now, or? Okay, I'm just asking if you have anything specific. So far, is everything making sense that I've said?
I know shit. Yeah. You need this trouble. And can you clear any doubts? Can you clear any doubts about the affinity curve?
Yeah, I don't mind.
Yeah, because people have a lot of.
Of course. That's a very. No. I'm not going to ask you about Trendelenburg's position. That was just for you to understand blood pressure in general. And by seeing how we treat it if we don't have adequate blood pressure.
So
We're not treating for shock, no, on the test. Although it would be cool, but I was using that to help you guys understand the temperature part of shock, as well as the perfusion part, the circulatory part, and then also the blood pressure part, and how they're all related. So when you have shock, to treat for shock, we give the trauma blanket to heat the body up. We give them oxygen, supplemental oxygen, and we put them in a position where their legs are above their head, so we get pooling of the blood. So now we have an increased blood pressure that's going to be good enough to deliver blood. So the reason why we do that is, generally speaking, our blood is going to have water, a buffer for temperature. And it's also going to be used, it's a dynamic system where if you're hot, you have vasodilation. If you're cold, you have vasoconstriction. And then, so now by giving them the trauma again, so blood can come in and deliver the oxygen that you just gave them. And the way that it's coming in is not only are you opening up the blood vessels indirectly, but you're increasing the force with which the blood is being delivered. So all of that together is going to help you to have perfusion, which is delivery of oxygen to the tissue through the blood. Did I lose you?
No.
Okay. So that was the whole point of that exercise. I wanted you guys to see that that basically, instead of just telling you blood does this, does this, I wanted you to see in real life why we need to know these things, because you could treat issues whenever it goes bad. Everything goes bad at some point for different people for different reasons. So you just have to be ready. All righty. Respiratory volumes. Leslie, by the way, did I explain the oxygen cascade good for you? All right, I'll wait for a response there. I know that there's some kind of delay. All righty. So I'm going to start with the prettiest diagram that we could use for this. And what I'm trying to show here is that they're all going to be combinations of the C. Am I back?
No.
Are you sure you're not? All right, cool. No, I mean, it's definitely possible. All right. So if you look at the IRV, VT, ERV, so all of these other respiratory capacities or volumes are just combinations of those first four. So what I mean by that, you can calculate any of the other ones, which are simply just adding different ones from those four. So the most important thing to understand is four, and then we can make sense of the rest of them. So from the bottom left, we have the IRV. Well, let's start with, sorry. Now I'm talking too loud, right? Am I talking too loud now? Yeah, it's pretty loud.
They can lower it.
Yeah, it's pretty loud.
It's fine.
I mean, I just fixed it, but sure. Okay. I won't argue with that. All right. So, so we have these four volumes. So your VT is the same thing as your TV. It's your tidal volume. Your tidal volume is the volume of air that you exchange with each normal, quiet breath. So this isn't a forced breath. This is just your normal breathing in and out when you're not even paying attention to your breathing pattern. So we call it the tidal volume because it has a tidal look to it. If you look at the small squiggles from the spirometer results that you see in the left lung over here, so these small squiggles right there, those are basically, they look like waves. So that's your tidal volume. And within our alveoli, we have tidal flow, where air comes in and goes around the spheres that make up our alveoli. When it circles around the spheres of our alveoli, right? No? One second.
This isn't the capillary around the alveoli.
What?
Am I wrong?
Isn't the capillary what?
Around.
Isn't it on the alveoli? Isn't it like?
Okay. So these are your alveoli, right? And then they're wrapped by these blood vessels that make up a bed. This is your capillary bed where you have the gas exchange. So you know it's a capillary because of the color and the fact that you're going from arteries to arterioles to the capillary bed to the venules to the veins. So you have an exchange of gas happening at the capillary beds. And because of their small size in diameter, they're going to be very thin, and therefore they're going to be easier to burst. So they're very thin because you need them for gas exchange. That's the pro. The con is by being so thin, they're going to rip. So that means that you need to make sure that you have something to collect the fluid that is leaking out, and that is your lymphatic system. It's collecting the fluid from the leaky capillaries.
God.
Okay. So within each of the air on the respiratory surface, by making sure that it's going to go around the alveoli. So it's
Happy and one of them confused.
Okay, yeah, you're totally. I could write everything.
yeah, just say okay, I'm reconnecting. I'm gonna have to wait. I might. Whenever the air comes in, because of the shape of the alveoli, instead of going through a straight tube, it's now to circle around the entire circumference of your alveoli, meaning that you have points of exchanging at each of those, at each point within that sphere. And then on the other side of the respiratory membrane, which separates your air from in your alveoli from in your capillaries, you are going to have these capillaries. And oxygen is going to be able to go from a place of higher concentration to a place of lower concentration, which is within your red blood cell. And they're going to be binding to the iron that's in each heme unit, which is in each of your hemoglobin, which is going to make up 99% of the volume of your red blood cell or your erythrocyte. Okay, so if anyone has any questions so far, please stop me here so that I would be able to explain that before we get to the next point, which is the rest of the capacities. Yeah.
So the cartoon that you showed me, one of them was happy and one of them was like upset. Doesn't mean like only one side of the lung is ventilated?
So one side is when someone feels it has oxygen, and then like in the capillary bed is where diffusion happens.
But isn't the alveoli like on both sides?
I have no idea why those face. Yeah, alveoli is definitely on both sides.
Yeah, like one of them is happy.
There's no reason.
Oh, there's no reason. Okay.
And in fact, your right lung. Oh, maybe that's why they made that joke. Nah. Your right lung is a little bit wider, the bronchioli that goes to it. So I don't know if they're trying to say that's why it's happier and it has air, but I don't think that they thought about it for any reason and said it. There's alveoli in both of your lungs, and both of them are going to inflate and deflate in a human that is with normal physiology.
Oh, this? Oh.
Did anyone say anything yet?
No. Melina retracted.
She retracted?
Yeah.
What does that mean?
Like she deleted the message that she wrote.
About wanting to know about tidal volume and.
Oh no. She said something.
She said the thing was lagging. She said,
Oh, okay.
is this camera on?
Is that camera?
No.
Jordan. You're just so annoying.
Alrighty. So
You're-
it's already back on here. Don't worry.
Yeah.
You guys got 30 seconds. Okay. So okay. So with your lung volumes. All right, guys.
No, go ahead. Continue listening.
Okay.
No.
It's on monitor. You're coming to its screen.
Okay. So back to business. So you have lung volumes and capacities. So we just said that we have tidal volume, which is related to tidal flow. And that's going to be the volume of air that you exchange in a normal breath. And normal breaths are going to be quiet, usually. If you have respiratory problems, such as an obstructive or restrictive disorder, then you might have a wheeze, in which case it's not quiet, or some other kind of sound. But for the most part, most of us are going to have quiet breaths, and those are going to be our normal breathe in, breathe out, and that's represented by the tidal volume. Now, the next volume is going to be the volume that we associate with when we take a deep, forced breath in. Okay. Okay. So you have your inspiratory reserve volume, your IRV, which is this big green portion, and it's exactly what it sounds like. So when you breathe in, that process is called inspiration. And then your reserve is the things that you're not using on a normal basis. You have it on reserve for when you need it. So the inspiratory reserve volume is the amount of space or air that you have in your lungs that you can use if you force an inhale. All right. So just so you guys know what's happening here. So now we have our expiratory reserve volume. And then that, and it's going to be the volume of air that you have on reserve for when you breathe out or expire your air with force. So forced expiration will cause you to use your expiratory reserve volume. Now, even after all of that, you're going to have some volume that is going to be left over. Your residual volume. This is the amount of air in your pink over here at the bottom that's going to stay there even after you force an exhale. So you're not going to remove everything in your lungs, even if you force your exhale. And in addition, there's something that's not even shown here, which is your anatomical dead space. So this is air that's going to be stuck in between breaths in the airways of your respiratory system. So within the conducting zone, you might have air that's going to go up and down but never leave or go in. And this is your anatomical dead space. Oops. All right.
So residual volume never gets used.
So residual volume, I don't know if it's never used, but that's the amount of because like the air is moving. it's not like it's a division in pink in your lungs where it's like, this is my residual volume container. Like, it's just like, these are just like how much it can be, basically. Like, this is saying it can be up to this much, and we're saying that the residual volume is what's going to be left after we take our forced expiration. And in fact, we could take it further by saying that our residual volume is equal to our TLC minus our VC, meaning it is equal to the total lung capacity, everything, minus the maximum amount of air that you can exchange in a single breath, which is your vital capacity. Your vital capacity, a living person, is exchanging what they normally exchange, their tidal volume, plus whatever they have on reserve to breathe in, their inspiratory reserve volume, plus whatever they have on reserve to breathe out, their expiratory reserve volume.
So we don't have to know inspiratory capacity?
We do, but I'm saying that your residual volume is equal to your total lung capacity, which is everything together, minus your vital capacity. And your vital capacity is everything except for your residual. So if you notice the colors, the combinations, you could piece together what's being added to come up with each of these capacities. So your inspiratory capacity is going to be equal to all the volume that you can actually breathe in if you try your hardest, meaning what you normally breathe in plus what you have on reserve. And then your functional residual capacity is going to be what you have left over after your inspiratory capacity. So if you take your TLC minus your IC, you end up with your functional residual capacity, which is what's left in your volume after you've breathed in everything you could breathe in. All right, guys, I'm waiting. Residual is the amount that doesn't reach the alveoli. Right.
Oh, we have another question. Are you ready?
What's up?
So Daniela can't post in the chat, so she asked me to ask you, please review Henry, Boyle, and Dalton's blog.
She can't post in the chat because... Huh?
Yeah, she can't post either. It says change the channel.
It's not letting me post.
That's weird.
No, it's
No, I believe you. I'm just saying that's weird. All right.
Oh, she doesn't have a YouTube channel. Haha.
Oh, I got you. Okay. So I'm assuming that's the same case for you, right?
Yeah.
Okay.
Before I forget, can we go over the QRS as well? When does the audio repolarization happen?
Yep, I got you.
I'll tell you that.
Okay.
So right now she was asking me about what? The gas laws?
Boyle and Dalton. Yeah, actually.
Yeah, I need to lower
That's
that.
like the best selfie light, bro. Oh, we about to take a selfie for a start.
No, you see my laughing class.
It's interesting. They're gonna fall asleep. Don't you see this group right here? Not pointing out people, but like, you know.
They probably hear you
I don't
too.
care.
You can't sleep, so we don't need to
Yo. Telling all these people that's here. Hell yeah, bro. Okay.
Okay, let's see.
Oh, bro.
All right, I'm trying to pull up a good image to show you guys all of them. What's up? All right. There you go. It's gone. No one can see you now. No one can see you now.
Next time I'll hold your ass. I'm like, yo, what you think?
Oh.
All right, 1 second. I'm pulling up. I was probably just going to use the one that I used in class, but I didn't know if there's a better one that could help to explain this. Just because it's very rare that people talk about Henrys. Even here. Okay. 1 second. Dalton, Henry, Boyle. Wow, this is actually probably the best one I've ever seen. I'm so happy about that. Man. Okay. 1 second. This is probably going to teach you better than I ever could. Oh, come on. Load. No, I just haven't switched over to it. It's not the delay with this system. But yeah, you could feel free to look over here. Okay. So gas laws are really talking about. Now we're not talking about Charles for respiratory anatomy as much. Don't really worry about that one. The ones that we focused on were these three over here. So Boyle's law is saying the more free space that I have in my subway cart for me to relax, the less pressured I feel. The less free space I have, the less space that I have to move around, the more compressed I feel. Compression is an increase in pressure, right? Compressed air. You're highly pressurizing air. Now, Dalton's is saying the total pressure of a gas is equal to the sum of the partial pressures of the independent gases. And it's on top of that, it's proportional to how much of them there are. So if you notice, there's more pink than there is blue. So it's not that you're just adding them up. This is going to have the highest partial pressure because it has the highest concentration. And furthermore, Henry's tells you the same thing, that you have the higher the concentration of a gas in a solution, the higher the partial pressure, and then it's also going to be related to the solubility of the gas in the substance that we're talking about. So meaning the concentration of that oxygen is going to be equal to the partial pressure times of the oxygen in the mixture of air times the solubility of oxygen in water, let's say.
Wait, can you repeat that?
Okay. So the concentration, how much oxygen gets dissolved in water, is going to be related to the partial pressure of the gas in the mixture. Because basically think about this in the way that he described it. You see that there's an interface between water and air. So air has more oxygen than water. So how does the oxygen go from air to the water? And why is it that some water has more oxygen than others? So Charles tells us that temperature and volume are related and therefore pressure inversely. And then from Henry's, we know that the amount of the gas that's dissolved in the water is related to how much of the gas there is in the mixture. So the partial pressure of the oxygen multiplied by how well oxygen can be dissolved into water. So if oxygen can't be dissolved, the concentration of oxygen in the water is 0. if it's very soluble, the more soluble it is, the higher the concentration will be because more of it will dissolve. Does that make sense? All right. All right, guys. So what's next?
PQRS. The PQRS.
The cardiac conduction system.
The people don't come here early. I think the earliest time we came was at 6:30. So fun enough. And the latest time we left was God, never left.
I've slept in this campus so many times over the years. So many times.
It's nasty, yeah. Sleeping in the library
Yeah.
during the final lecture is nasty.
Yeah, but it helps so much because,
I took brain mind experience, right?
we were in that together.
And I went to class literally 3 times. The first day of class, the second day of during midterm, and then the third time was during finals. And I learned the whole semester worth of shit in one night. In one night. 8 books, bro.
Okay, so the best source that I would say for this stuff is to go to that Aaliyah medicine video that I showed you guys in class, and just watch it in animation so that you could get it clearly. But before we go over the PQRS T waves, let's talk about the intrinsic cardiac system, which means it's the way that we are able to conduct electricity from the heart by the heart. So what I mean by that is that you have autorhythmicity. This is a property that only your cardiac muscles are going to have. And what this means is that your pacemaker cells that are found in your sinoatrial node, your SA node, which is found in the top right of your right atrium. So that's over here. And I actually made the mistake right now of saying left instead of right just because I'm looking at it and it's on my left. But remember that this is about the patient, not about you. So this person who we just saw in the cadaver lab has their sinoatrial node in the top right of your right atrium. And this is going to have your pacemaker cells. And what's happening here with your pacemaker cells, wait, can you please repeat the difference between residual and dead space? I think I missed that.
Because lagged out probably.
Ah, man. Okay. So residual volume, Cynthia, is what is left in the lungs after the maximum amount of air that you can exchange in a single breath. So think about that. I normally can breathe in like this. And then if I really want to, I can forcibly inhale and forcibly exhale. So by using the tidal volume and using my reserve volume for my inspiration as well as the reserve volume for my expiration, I've used the maximum that I can exchange in a single breath. This is my vital capacity, the maximum a living person can exchange in a single breath. But then you also have what's left over from your total lung capacity. What I mean by that is you can't move everything in a single breath, no matter how hard you try. So what's left over is this residual volume. And your residual volume is equal to your total lung capacity. Oh, I'm sorry, I'm pointing and I'm not showing you guys the screen. Sorry. So your residual volume is going to be equal to your total lung capacity minus the maximum volume of air that you can exchange in a single breath, your vital capacity. So you have this over here, that is your residual volume. Dead space is going to be air that's stuck in your anatomical structures of your respiratory system. This is the volume of air that might be in your conducting system, conducting part of your respiratory system, but it's not eventually, but not with a single breath. So it might take many breaths. Anatomical dead space. So to give you a visual, on that. You know what? I'm just gonna use this. Nope. Okay. All right, so I'm gonna switch over to the image now. So your total dead space is everything that's stuck in the anatomical dead space, plus your alveolar dead space, plus the mechanical dead space. And what's meant by dead space is that this is a place where air is not being exchanged in a respiratory function. Sure.
Okay.
Thank you. So residual capacity is equal to TLC minus VC. Absolutely. The respiratory zone in the respiratory system starts after the terminal bronchioles. Yes. So a better way to remember this is by using the words. So your conducting zone terminates at the terminal bronchiole. Your respiratory zone starts at your respiratory bronchioles. And I have to get back to your question about the PQRS.
Yeah, and I have another question.
Okay.
It's about respiration.
What was I gonna say? Respiratory. Man, I totally blanked on what I was about to search. Okay, so dead space, respiratory volumes.
You were talking about where respiratory system stops.
Conducting zone versus. Okay. So by looking at this diagram, you should see that this is the conducting zone where you're conducting air to the respiratory surfaces, which is your alveoli. Now, I never mentioned this in class, but just so you know, and it helps with your understanding, you have a force that's pulling your air in, and that is your negative pressure of your ventilation. What I mean by that is because of the pressure difference between the air and the inside of your lungs, air is going to want to go down its concentration gradient or pressure gradient. And so it's going to go from the outside air with the higher pressure to the inside of your lungs with the lower pressure. So because of that, you will have a sucking or suctioning, like a vacuum of air into your lungs that's caused by a pressure difference. So this is a negative pressure breath, which is what people normally do. So conduction is moving the air down that pressure gradient to the place where you have respiratory exchange of gases. So that means that you're going to be able to suction the air through there or conduct it through. And this is an example of bulk flow, because you can't diffuse the air through, it wouldn't be efficient. So you're going to move it along this highway of air to get to the point where you get to your local roads, where you can make local deliveries, house to house. And this is where you have the end of the conducting zone, the beginning of the respiratory zone where you get diffusion. And in between that, you have your bronchioles that mark the end and the beginning. So this bronchiole over here that marks the termination of your conducting zone is your terminal bronchiole. This bronchiole over here that marks the beginning of your respiratory bronchiole is going to be your respiratory, your respiratory zone is your respiratory bronchiole. No problem, Cynthia.
So why do we breathe out?
So for the same reason that we breathe in, but breathing in is an active process. To breathe in, you need to be able to contract your diaphragm, a muscle fiber, to pull down and open up your thoracic cage, increasing the volume, which changes the pressure in your lungs, causing it to go from high pressure to low pressure. As it fills up, the volume of free space in your lungs starts to decrease. They start to get crammed up in there to the point where you're equal to the outside air. So the difference of outside minus inside is 0. So now your contracted diaphragm relaxes and you get elastic recoil of your lungs. So goes back to its original position. And because it did that, now the volume has decreased. And you already filled up your alveoli with air. So now already you're going to have a huge buildup of pressure within your lungs, so much that the pressure is now higher in the lungs than the outside air. So now it's going to be a release of the pressure from the outside to the inside, from high to low. And the pressure volume loop continues to loop, keeps happening. And that's how you go through the whole cycle of a respiratory breath in and breath out. Yeah.
Okay, so you lost me at the whole pressure thing, right? So you breathe in, diaphragm contracts, right? It goes down. So thoracic cavity opens up, right? Air goes into the lungs. So that means the pressure is what? Higher or low?
Where? In your lungs?
In your lungs, right? Because outside is high, so high to low.
Exactly. Then while you're breathing in, the pressure increases in your lungs as well.
Yeah.
And when it's equal to the
Then
pressure outside,
So 0, right?
0, and then and then your diaphragm relaxes, and the recoil
Goes
of
up,
your lungs,
air comes out.
and then for the same reason.
No, I get it. It's okay.
Now, the average breath is describing happens a lot quicker, right? So imagine with this model. You have your diaphragm over here. It gets pulled down, causing the increase in the amount of free space. So that means that it's under less pressure. This would be so much helpful if it actually worked.
Thoracic cavity opens, pressure inside the lungs
Starts increasing.
increasing. Once the pressure in the lungs is increased and with the outside air is the same, so it's zero.
Yeah.
And your diaphragm relaxes, goes back up, the air goes out.
Yes.
Okay, yeah, we're good.
Yeah, but did you memorize it or can you visualize it in your head? You understand?
No, I get it like here, right? So you're breathing in, okay, your diaphragm has to contract so your thoracic cavity can open so your lungs can inflate, right? Or else
Exactly.
lungs can go.
And then the volume inside the lungs
It's increasing.
starts getting cramped up.
No, pressure increases,
Yeah.
volume decreases.
Volume decreases because of all that space that the air is taking up.
Cool.
She gets it, bro.
No, I promise I get it. If I didn't get it, I'd ask you to explain it.
Yeah.
You know?
I got you. Actually, now that I think about it, okay. 1 second. Ventilation.
So ventilation is the process
animation.
of breathing, and respiration is what actually goes on in the lungs, the gas exchange, blah, blah, blah.
Correct. Oh, I should have clicked on that other one. All right, whatever. We'll use this.
Mechanics of breathing.
Can you guys hear it?
Consciously take a breath and think about the fact that there are 10 different muscle groups working together to make these muscles. These muscle groups include the diaphragm and intercostal muscles. The main muscle used for breathing is the diaphragm. The diaphragm is a dome-shaped structure consisting of several large muscles, which is sandwiched between the chest cavity, containing the lungs and the rib cage, and the abdomen, containing the digestive system, including your stomach. The muscles that move the rib cage itself are the internal and external intercostal muscles. They are each attached to the ribs and run between them. To inhale air, the diaphragm contracts and moves down, while the external intercostal muscles contract, forcing the rib cage up and out. The combined effect of the diaphragm and intercostal muscles increases the volume of the chest cavity and expands the lungs. This expansion of the lungs increases their volume, reducing the pressure within them, causing air to be drawn in. This action is similar to a piston sucking petrol into a car engine. In normal breathing, we use around 25% of our lung capacity, which is called our tidal volume. As you inhale deeply, the diaphragm moves further down into the abdomen, pushing your belly out, giving more room for the lungs to expand and draw in more air. This type of breathing is called belly breathing or abdominal breathing, and is critical to prepare you for your free dive. Belly breathing allows you to completely fill your lungs with air. The maximum amount of air your lungs can hold is called your total capacity.
1 second. I'll give you. I wasn't too happy with that one. We'll use the McGraw-Hill one.
At the end of expiration, barometric air pressure and alveolar air pressure are equal. Therefore, no movement of air into or out of the lungs takes place. Inspiration begins with contraction of inspiratory muscles to increase thoracic volume. This results in expansion of the lungs and an increase in alveolar volume. The increased alveolar volume causes a decrease.
So you see how now this P alveolar, which is equal to -1, is actually related to, that's -1 is compared to the outside air. So it's just a single millimeter of mercury less than your outside. So instead of 760, it is now 759. Or if we were just going to use oxygen, you have a difference of 160-ish versus 159. So now it is just one less than the outside, and that's enough to allow the volume to come in. Yeah. Usually it only changes by about 2 up and down.
In alveolar pressure below barometric air pressure, and air flows into the lungs. At the end of inspiration,
No problem,
the
Daniel.
thorax and alveoli stop expanding. Airflow into the lungs causes alveolar pressure to become equal to barometric air pressure. Because the pressures become equal, no more movement of air occurs. During expiration, the volume of the thorax decreases as the diaphragm relaxes and the thorax and lungs
see how
recoil.
the volume of the thorax
This results in a decrease in alveolar volume and an increase in alveolar pressure. Since the alveolar pressure is now greater than barometric,
changing the pressure, causing you to breathe out.
Okay, see, I remember the other way. I remember you breathe out, so the diaphragm goes out.
Okay.
Air pressure, air flows out of the lungs. Air continues to flow out of the lungs until alveolar pressure becomes equal to barometric pressure.
Is it over? No, almost. No, I didn't freeze it. It was just the end of that video. Okay. Alrighty, so hopefully that answers your question. So now let's get back to this whole conduction system. Yeah, so we were here. So we have this conduction system that we're taking a look at, and you basically have your sinoatrial node, your SA node, that, as the name suggests, is found in your atria, specifically the right atria, and it's responsible, as the name suggests, for producing the sinus rhythm. So the sinus rhythm is this 60 to 100 beats per minute that everyone who's healthy has. So the pacemakers that are found here are going to create or generate action potentials in response to calcium influx that are going to, like what's happening in your nervous system, an action potential is going to propagate and send a signal. In this case, the signal is contraction of your muscle. Which muscle? Your cardiac muscle. So you're going to have the signal pass from the sinoatrial node to the next place, which is where it's going to get to the atrioventricular node, which is going to be the node that is in between your atrium and your ventricles. And now it's going to travel from the AV node down through the AV bundle, your atrioventricular bundle, which is found around your interventricular septum, which is a fancy way of saying the division between the two ventricles, because internet between computers, this is interventricular between the two ventricles. And just like your nose septum that you get pierced, this is the septum between the ventricles. It's the division between left and right. So as it passes through the interventricular septum, it's now going to continue. And these bundle branches will then become what we call your Purkinje fibers. And fibers are just nervous tissue, right? So some things to note about this. We said your sinus rhythm is created by your sinoatrial node, where you have pacemaker cells. So now, how does that signal that's generated here through action potentials spread through the heart? Is AV bundle equal to the bundle of His? Yes. So your sinoatrial node is going to have these pacemaker cells that are autorhythmic. So every time that it generates an impulse, it creates a coordinated contraction of your tissue, your muscle tissue of your heart, your cardiac tissue, which we also call your myocardium. So the reason why it's coordinated is that you have tight gap junctions and intercalated discs. intercalated discs are these little joining features between adjacent cells, just like your tight gap junctions, that allow for all of the tissue that's connected in this one synctium, which is a whole unit that beats together in sync. So your heart has two of them. Your atrium together beat in one synctium, your ventricles beat together in a second synctium. So everything in that synctium will beat together because the wave of depolarization from the action potential is going to spread from cell to cell of these myocardium through the intercalated discs. So the reason why we have a second synctium, which is slightly delayed, is to allow for the atria, your left and right atrium, to first contract and leave a delay before your ventricles to contract. Now, this delay is as little as 1/10 of a second. Your lub-dub sound is going to kind of be related to that indirectly. So what's happening here is that your atria are beating together in coordinated synctium, and then that's going to force blood from the atria into the ventricles. Once they get into the ventricles, there's the delay while it's filling, and then the ventricles will beat. But you need time, a little delay, for the ventricles to get filled before they eject their contents. Otherwise, they're not pumping anything. What an inefficient use of energy. So they're going to wait for it to be filled so that they have a higher stroke volume, which is going to be related to your cardiac output, which is how much blood that you're moving in a single minute. It's equal to the times that you beat per minute, your heart rate, multiplied by the volume of blood that you're moving with every beat or stroke of your muscle. So your heart, although it has very impressive stroke game, should not be stroking at a rate that is so high that it's not able to actually pump anything and just wasting energy. It's not going to jackhammer. So after your initial filling from the atria contracting together, you are going to have your ventricles have an isovolumetric contraction. This is the point where you have a contraction of the ventricles that does not lead to any change in volume. Isovolumetric contraction. So why would we do this? Seems like a waste of contractile force. Well, actually, we're building the pressure. The pressure that we need to be able to propel the blood, not just out of the ventricles, but through your entire system or your pulmonary circuit. So during this time of your isovolumetric contraction, you're going to have a huge buildup of pressure because the muscles contract, meaning there's less free space, meaning the pressure in here and in here is going to go up well, not exponentially, that's a bit of a hyperbole, but it's going to increase to a point where you have a high enough gradient that allows for blood to be ejected with force and go through your pulmonary artery or arteries rather, or your aorta to the rest of your body. Okay, one other thing to take away from this image. You have your heart, because it's so important, has a bunch of backups. It's a redundant system. So what this means is that it has a lot of things that do the same thing. redundant. Fail to create a rhythm because of a block, whatever it may be, that's causing it to not be able to do it, then you're going to have your AV node take over. If your AV node fails, then the bundle of His will take over. And so you're going to have it decrease in efficiency because this isn't your first string. These are the people off the bench that you're calling to do the job. But they'll still be able to play. So this is the backup system so that if your SA node isn't working, you have, instead of 60 to 100 beats per minute, you end up doing 40 to 60 with the AV node. And if the AV node doesn't work, then your bundle of His will produce your 20 to 40. And if that doesn't work, you're dead, producing 0. So, all that to go back to this one diagram where you're looking at the electrical waves of the heart. But if you don't know the system, the wiring, and what's happening because of the wiring, it makes no sense to describe the EKG. But now that you do know it, so your P wave is associated with the depolarization wave that starts from your sinoatrial node and spreads to the left and right atria. As it's completing its spreading, you get this PQ interval. This is all going to be the prelude to your QRS complex, which is this huge spike where a lot of things change at once. And what's happening in that Q region is that you have atrial repolarization that's going to start. But that's going to be masked by the fact that in your QRS complex, you're actually witnessing the spread of the electricity down through the interventricular septum to your ventricles, and now you are starting to see the rapid depolarization of your ventricles, which is this huge QR depolarization that you're seeing. And then finally, you are going to reach this point where you actually reach a complete ventricular depolarization, in which case the electrical signal isn't spiking anymore, but now has become stagnant. It's ST. So now after that, you're going to have repolarization of your ventricles, which is your T wave. And once that is done, you have the tail end of your T wave being shown over here for ventricular repolarization becoming complete.
So when does arterial repolarization happen?
So following this diagram, yellow is depolarization and red is going to be your repolarization.
So QRS.
So during this QRS phase, in that beginning part, this Q drop, during that whole part where the signal is moving down, this is already starting to repolarize, because after the action potential passes, you shoot up, you go down, and then you start to come back to your resting membrane potential. So you have your refractory period ending, right? The refractory period is the part of the time where you have to be in a rest mode, right? So while these are resting, they're being repolarized. That's the signal, which was delayed by 0.1 seconds or so, is already starting, or less actually, is already spreading to your ventricles and starting to cause the contraction of your ventricles.
Negative.
Why?
Okay, so while you have your atria depolarizing, while this is happening, your ventricles are at rest because they've just gone through their action potential. and they're all beating at once. So really, all you have to think about is simplify this in your head to 2 synctiums. You have an action potential in one that sends the signal to the second, and then that sends the signal back, and like it just keeps going back and forth where you have one synctium beat together, both of your atria, followed by both of your ventricles. And while this is beating, this is opening. So that now it could refill, so that whenever this opens, this closes.
Okay, yeah, okay.
Right? So in order for that to happen, it has to go through the cardiac cycle, and you're going to have this whole loop happening where you have 4 distinct points. And
wait, and who said that arterial polarization happens between RS? I heard it from somewhere.
it still would be happening during that entire time, but the QRS complex is so much greater of a change of that can be recorded that actually it completely overshadows what's happening during your
Wait, so what's R again?
that huge R is your ventricles contracting. Okay. So just like you have a pressure volume loop for your lungs, your pulmonary system, you have a pressure volume loop for your heart. And what this means is that you have 4 different points of your cardiac is you're going around this entire loop, which is why it's called. So let's go over the different things this loop can tell us, and then we'll go through the points. So by going between, so on the x-axis, first let's start with the axes actually, I'm sorry. On the x-axis, we have the volume that's found in your ventricle. On the y-axis, you have the pressure that's found in the ventricle. Remember that we're only looking at the ventricular part of your, wow.
And if you scroll up, there's
more. Oh, no.
Yeah.
I lost everyone, huh?
Yeah.
Okay. We'll get to that in a second. Okay. So there are 4 different distinct points. And what we're seeing over here is this is the maximum volume that the ventricles are filling to. And this is the minimum volume. So what that means, if this is the maximum volume, that means that this is when it's the most filled. And this is where it's the most empty.
So that's atria closed, ventricles most filled.
Let's just focus on the ventricles right now.
That's not
I'll get no, you're right.
Okay.
But just follow this first, and then I'll answer your question. So you have the total volume, the max you can get, and the minimum. And if it's at the max, it means it's been filled. And if it's at the minimum, it means that it's empty. So we have 2 parts of our blood pressure. At the point where the ventricles are contracting, you have your maximum force, your systolic blood pressure, the 120 of the 120 over 80. And then you have the point where you have the relaxation, where it's filling, and that's going to be the 80, the bottom number of your 120 over 80. So your heart is going to be in diastole, the relaxation phase, for most of the time, because that makes sense. It wants to only work when it's efficient to work. So 2/3 of the time it's relaxing. How nice to have this leisurely schedule, right? So it has a 9 to 5, it sleeps the rest of the time, and it chills the rest of the time in between when it's sleeping and when it's working. So just like a standard human. So now that we know that, okay, 1 second, Malia. We'll get to all of your questions. Okay, so this part where it's at its maximum is going to be when it's most filled. It's going to be most filled at the end of the time where it's being filled, your end diastolic volume. And it's going to be the most empty after it's already contracted and ejected all of its fluid, the end systolic volume. So now you can measure your stroke volume based on this pressure volume loop by subtracting the end diastolic volume from the end systolic volume. Because this means that if this is how much was in it at the end of filling, and this is how much was in it at the end of emptying, that means we moved this much in a single stroke of the heart. Hence the stroke volume. Okay. Now what makes it so that we have the pressure to be able to do that? So let's look at what's happening in our diastole versus our systole. So at the end of our diastolic, in the end of diastole, we're going to have the most volume, and now we're going to have an isovolumetric contraction, meaning the volume, 120 milliliters, never changes during this whole contraction, but the muscles are contracting, meaning that there's less space inside the ventricle, which means, according to Boyle, that we are going to have an increase in pressure. So now the increase of pressure is enough so that it is going to be greater than the pressure of resistance that's coming from the narrow blood vessels who are going to use this pressure gradient to have the emptying or ejection of your ventricles. And it's going to happen in a way so that you actually have an emptying of your ventricle. You go from having 120 milliliters to having 50. So after that, you're going to have an isovolumetric relaxation, where the heart, where the volume of your ventricles is not changing in terms of how much blood it has in it. So instead, it's going to stop being contracted, which is going to give the space for the blood, which is going to decrease its pressure from 100 millimeters of mercury down to 20, 30, whatever this is, 10. So these are the 4 points of your pressure volume loop that are happening at all times in your cardiac cycle. Yeah. So now let's get back to all your questions with the electricity. So that was your plumbing. Let's get back to electricity. You good? Okay. So let's see, what are the questions? 1 second. Q wave corresponds to depolarization of the interventricular septum. Yeah, it does. Malia, so for that, yeah, it is. Giorgio, can you clarify if you ask where atrial repolarization occurs? If it's during the ventricular depolarization, which is QRS complex, or during ST. So your repolarization is going to happen. So look over here. This is we're right here, we're looking at the electricity of the heart in terms of its action potentials. So we go from being at rest. Oh man, 1 second. Okay, so we go from being at rest, we're looking at the action potential in your heart. And then the action potential is generated. So this over here, you're going from -60 towards 0. So depolarization by definition means that you're losing the difference between the across a membrane. So that means that the inside and the outside are becoming very similar. So the difference between the in and the outside, or the membrane potential, is going to be closer to 0, which is why you are moving from a point of being at -60 over here to getting closer to 0. You are depolarizing. And in fact, usually you will cross over the 0 point and have an overshoot before repolarization happens, which is the downward slope over here. So huge difference. So you want to make sure that the inside minus the outside, meaning there's. Okay, so during this repolarization of your atrial muscle, right now we're looking at the SA node, let's go down to atrial muscle. So this part is rapid depolarization. After you become depolarized, you have the repolarization. Repolarization, as you see, is happening during the same time that your ventricles are in their resting membrane potential. And at the very end of repolarization of the atria, you have depolarization of your ventricles. And as your ventricles are repolarizing, you have your resting membrane potential happening in your atrial muscle. So, syncytium, boom, 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 boom. These two beat together, these two beat together.
Is that the lub dub?
The lub dub is the sound that's happening when you have your isovolumetric contraction. So the blood is going to be squeezed against your valves. And because the valves are closed, but you have the blood rushing into this against a closed valve, it's going to crash against the valve. So instead of having laminar flow, where something's moving like a lazy river, it's just flowing, you don't hear a lazy river. You get it crash like a wave against your valve, which means that it is turbulent flow. We can only hear things where you have turbulent flow, like your oceans. The more turbulent the body of water, the louder the sound. Which is why we hear the lub dub, which is the hit against your AV valves versus your no, just your AV valves. Sorry. No, but wait, why is it lub dub? AV valves followed by your semilunar.
Yes, okay.
So there's a certain point where all the valves are closed, and that's during the emptying over here that we're seeing. So during the emptying phase, while the valve is closed, you're going to have one of the.
My voice would have been down by now. Yo, he spoke.